All right, so I'm going to try and make a short video here to complete your notes for section 3. Hopefully you've already started these things because you were in class. If not, I'll run through some stuff real quick. But there was also another video that I posted. Make sure you watch that first. It actually does have more details in it than we're going to go through here. They talk about a primer and some other things that are in this DNA replication process. But the book that we use actually simplifies things a little bit, which is good because then it's easier for you guys to process. But understand, even the stuff that we're going to go through is a little bit complicated, the leading and lagging strands. So we're going to summarize that process of DNA replication and the role of enzymes in that replication. So you should already have this blank filled in. We answered this question in class, so I can walk through it real quick, right? Replication is when DNA is copied. DNA is copied during that synthesis part of the cell cycle. Remember, there's G1, synthesis, S, mitosis. Mitosis is broken down into prophase, metaphase, anaphase, telophases, and cytokinesis. But otherwise, you also did some base pairing here, right? You guys already know how to copy a piece of DNA because A goes with T, G goes with C. So as I go through here and just base pair, you can do this just as quickly as I can, and you probably did it faster when you were in class. But the thing that we added that's a little bit different, right? We're going to talk about how there's a directionality to each of these strands. There is a direction in which they're built, 5' to 3'. So those labels do matter, and they're always opposite. If one strand is running this way, the other one's running that way, right? So that's going to be a little detail that makes for a big problem when it comes to leading and lagging strands, but we'll talk about that as we get to it. So we're going to try and go through this replication process quickly, right? I'm going to draw a picture for it. So I have to have a piece of DNA, so I draw a helix with this big bubble opened up in the middle. Because I should have already had, excuse me, that was looking weird. I should have already had some helicase molecules coming through here and opening things up for me. So helicase is this enzyme that's going to be right here at these two replication forks, we call them, that is just going along and unzipping that double helix. And you can think about this pretty much just like your book bag, right? You got two zippers that you can pull to open it up, right? You're just opening this space in here that exposes bases that you can start to base pair with. So as long as I got this replication origin started, right, DNA polymerase should be able to come in and start base pairing and making new strands of DNA. So I'm going to start with a DNA polymerase somewhere in the middle here, but I also need some labels first, right? This black strand, right? If I just arbitrarily mark this as 5', then I'm going to follow my little loop-de-loop here. That means this side's going to be a 3', right? So again, this was 5'-ish. This is 3' for that original strand. So it should be the opposite for my other strand. This is 3'. Again, should probably put a 3' here, just so I can see that. 5' here. And then all the way through, both strands are running opposite of each other. Again, that direction does matter. And it matters because polymerase builds from 5' towards 3'. So if I start with a 5' here, and this new strand, this purple strand I'm going to draw relative to the old strand, it has to be built in this direction towards my replication fork, because that's my 5' end, and I'm building towards what ultimately would be a 3' end over here. And this strand actually is going to be my leading strand that they talked about in the video, because it can continuously be built as it follows behind your zipper, right? As this continues to unwind, and I'm just going to draw it continuing here. I'm going to build all the way to the end over here, which would be my 3' end, and that strand should be built continuously. But that means that if I started on this side, and I'm going to build from 5 prime to 3 prime, if I start with 5 prime right here in the middle, 3 prime's in this direction for this strand, right? So I can build this new piece continuously, following right behind the zipper again, all the way to the end to where my 3 prime label would be. But that leaves half the DNA unfinished. And that's still going to be replicated, and it has to be replicated by polymerase. But essentially, I'm limited, right? I can bring in a polymerase right here, but I'm only going to be able to build so much of this new strand, right? Excuse me, I wanted to change to the other pen, right? So I'm going to build a piece of DNA that I'm using a different color for a reason, and I'm going from 5 prime towards 3 prime, but I can only build so much before I've already done the rest of that, right? But also I have to wait for this to unzip and unwind a little bit further before I can build another little chunk, maybe from here to here. And these are what we call our Okazaki fragments, and those make up our lagging strand of DNA. Again, because helicase opens up this replication fork, and I can only build DNA in one direction. I can build it this way on this strand. That means that I'm going to end up having to build this other side piecewise. I built the purple side continuously, but I can only build the pink portion of this DNA in fragments. And again, that has to do with that anti-parallel. It has to do with the fact that strands of DNA are running in opposite directions to begin with. And it's a limitation of our polymerase. It can only build from 5 towards 3. So again, that's kind of a complicated thing. Hopefully that other video also helped you understand that, but we'll talk about this again to make sure we get it. But I'm going to have these leading and lagging strands that I'm dealing with. Again, I made lagging strands pink. I made leading strands purple. But polymerase is just base pairing as it goes, matching A's with T's and G's with C's, and bringing in new nucleotides, linking them together, but they only can link from 5 to 3. Polymerase can only link from 5 to 3. So I end up with leading and lagging strands. Most complicated part of this section by far. Otherwise, the last thing that's going to have to take place is I'm going to have to link those strands together, because it has to be a continuous piece of DNA when it's all said and done. So I'm going to bring in a ligase, right? So eventually I need another enzyme to come in here. We're going to call that ligase, and it's going to link this strand to this strand to make it continuous. So it's one continuous piece, even though it's different colors still, right? But there are lots of different places I have to link things together. Every one of these fragments has to be linked to the next one. So I got lots of places in here where I have to provide that final connection, and ligase is going to do that job every time. So now I have this continuous piece of DNA matching up with an original piece of DNA. That's the other part here, right? It's supposed to be a semi-conservative replication because I have one new piece of DNA, right? And again, I use a little bit of pink and purple, but combine that, you get that new DNA. And the original DNA molecule was just that black that I had to begin with, that was separated and I started replicating off of. So again, this is an oversimplified picture of a very complicated process. So hopefully you're starting to understand it. Other little details. Enzymes usually kind of tell you what they do in the name. So that DNA polymerase builds the DNA polymer, obviously, right? All enzymes end with that same ending. So I have that ligase, and that ligase is supposed to link those fragments together. So usually you can kind of tell from the name of an enzyme what it does. The helicase, right, helps to unwind that helix structure. the double helix has to be separated in order to do all of this. So just think about that when you see your enzymes. Otherwise, we also talked in class about how this replication is remarkably fast and accurate. You can replicate your entire genome in a cell in a matter of hours. It's ridiculous how fast this can go, right? So when we were doing our base pairing at the beginning, I could finish this strand in, 5+ seconds, but your DNA in your cells is being copied at up to 50 nucleotides per second. You're going to start at many different places at the same time so you can get it done as fast as possible. Prokaryotes, it says, they only start from one place. So again, I had that replication bubble over here. I'm saying that in your DNA, there would be multiple bubbles opened up and down that chromosome just to make things a little bit faster. You can get done faster. And that makes sense. It should be logical. But if it was a prokaryote with circular DNA, you can just go around in a circle and you're done, right? So it doesn't really have to start in multiple places because the DNA is short enough and it's circular that you can just go around and finish just like that. But otherwise, it's remarkably fast, right? Replication is really, really, really fast. It's also super accurate. DNA polymerase can proofread as it goes, meaning that the errors, you're only going to have one wrong base in about 1 billion base pairs, and even those get fixed. So the equivalent of that, I saw that in a different video I was watching, is you could type at 60 words per minute for like 20 years or something weird like that. And I'm talking continuously, so no sleep. You just got to type for 20 years straight at 60 words per minute. You'd only make one error. That's what this is the equivalent to. And that's just insane how accurate that is. But it makes sense because you can't bring in the wrong base. They don't fit, right? So you can't really make a lot of mistakes. The other weird thing I found out the other day, right? Your DNA is already replicated trillions of times since you were a single cell. In your lifetime, you're going to create 1 light year of DNA. Now, a light year is a distance, just in case you didn't know that, and it's the distance it's the distance light travels in 1 full year. Right? It takes 8 minutes for the light from the sun to get to our planet. Imagine how far light travels in 1 year, right? That's how much DNA you're going to make in your lifetime, right? So this is just amazing, right? When we talk about how elegant and impressive that DNA is, that molecule that makes you everything that you are, right? We're not joking around, right? It can do a lot of impressive things, and you're going to do a lot of that in your lifetime. So hopefully that's all you need. Should be good enough for notes.